Hey everyone, and welcome back to The Explainer. Today, we are diving into a really fascinating mathematical detective story. We're going to unravel the inner workings of linear mixed models. Now look, I know what you might be thinking right off the bat. Heavy statistics, confusing algorithms, a bunch of dense matrix algebra. But seriously, don't worry. We're going to demystify this entire framework step by step, totally visually, and without getting bogged down in a million equations. We really just want to see how this incredible mathematical tool solves messy, real-world problems that standard statistics literally just can't handle.
Okay, let's jump right into our roadmap for today. First, the cystic fibrosis challenge. Second, why standard regression fails. Third, we'll introduce linear mixed models. Fourth, fixed versus random effects. Fifth, serial correlation. And finally, estimation using ML and REML.
Starting with Section 1, the cystic fibrosis challenge, a medical mystery in longitudinal data.
To really understand why we even need this advanced math in the first place, we have to ground this in a high-stakes problem. So our anchor for this explainer is a real clinical dataset from a 1997 study published in the Journal of Pediatrics by Davis. The researchers here were hunting for answers to two massive scientific questions. First, what is the average rate of decline in lung function, which they measured as a percentage called FEV1? And second, is that rate of decline different for patients who carry a specific genetic mutation, specifically those homozygous for the F508 mutation? You know, these aren't just abstract numbers on a whiteboard. Pinning down these rates is absolutely crucial for real patient care and treatment planning. And to get those answers, the researchers couldn't just look at a handful of people. They had to track a pretty significant cohort over a long period of time. We're looking at exactly 200 subjects in total. Think about that. That's 200 individual humans, each with their own unique medical journey, unique genetics, and their own baseline health, all coming into the clinic for checkups over many years. But here is where the messy reality of clinical research hits us right in the face. Real life doesn't run on a perfect schedule, right? The data is incredibly clustered and uneven because patients just don't show up to the clinic like clockwork. Look at this breakdown. You've got 49 subjects with exactly 6 check-ins, 52 had 7 check-ins, 36 people came in 8 times, and 63 subjects managed 9 observations. So what we're left with is this highly unbalanced longitudinal dataset where everyone is just on a completely different schedule.
Which brings us to Section 2, why standard regression fails with these uneven observations.
Now, what's really interesting about this slide is just how glaring the problem becomes when you visualize it. Faced with all this uneven data, you might be tempted to just, throw a standard linear regression at it and draw a line of best fit. But look at all those giant empty NA spaces in this wide data array. This is what our dataset actually looks like when we align patient check-ins by age. Subject 1 totally drops out after age 14. Subject 2 doesn't even enter the study until age 12. And Subject 3, they only give us 3 data points late in their teens. The thing is, standard regression requires every single measurement to be completely independent. But obviously, patient 1's lung function at age 10 is highly correlated to their own lung function at age 8. If we just ignore these blank spaces and pretend every measurement here is an isolated, independent event, we completely violate the core rules of statistics. The math just falls apart.
So, moving to Section 3, enter linear mixed models, combining random effects and serial structures. Okay, so we have this clustered, uneven, highly correlated mess of data. To actually make sense of it, we turn to a really elegant mathematical framework heavily formulated by statisticians Laird and Ware back in 1982, the linear mixed model. The whole genius of this model is perfectly captured in this core quote. A linear mixed model combines random effects and serial structures for clustered and nested data. See, instead of trying to cram our messy reality into a rigid traditional box, this model handles it by splitting the math into 2 distinct parts. You have a mean model that captures the overall big picture trajectory of the disease, and a covariance model that specifically wrangles all that individual variation and the awkward timing of the check-ins. Let's break that down in Section 4. Fixed versus random effects, averages versus individual trajectories. To really grasp how this model works its magic, we need to understand this dance between fixed effects and random effects. So the crucial point is knowing the difference here. Fixed effects represent the systematic variation. Think of this as the population average. It asks the big question. Across all 200 patients, what is the overall average rate of lung function decline? It's the signal we're looking for.
On the flip side, the random effects handle the random between subject variation. This allows for entirely unique individual trajectories. It mathematically acknowledges that, hey, patient A might just inherently have stronger lungs than patient B, and that is completely independent of the overarching disease trend.
And the incredible thing is, we get to dial in exactly how much individuality we want the model to accommodate. If we just tell the model to use random intercepts, it basically says, okay, every patient starts at a different baseline level of health on the graph, but let's assume they all decline at the exact same parallel rate. But if we use random intercepts and slopes, we give the model maximum flexibility. Now it says every patient starts at a unique baseline level, and every single patient has their own completely unique angle or rate of decline. It essentially filters out each patient's individual quirks so we can cleanly see the actual signal we're trying to measure.
Next up, Section 5. Understanding serial correlation and the influence of time.
So we've accounted for individual baselines, right? But we still have one more major hurdle to clear, and that's time itself. Even if we map out a patient's unique trajectory, measurements taken from the exact same person still aren't independent. And this brings us to a phenomenon known as serial correlation. It's simply the concept where observations that are close in time are much more strongly correlated than observations far apart in time. I mean, think about it. A patient's lung capacity today is obviously going to be super similar to what it was just a week ago, but it'll be way less correlated to what it was, say, 5 years ago. We absolutely have to give our model a mathematical way to understand this fading influence of time. And this brilliantly illustrates how we pull that off. Diggle's 1988 mixed plus serial model breaks down a patient's variance into 3 highly intuitive biological sources. Step 1 is the random intercept. Think of this as the patient's trait, their inherent steady-state baseline. Step 2 is the serial variation. This is their temporary state. So, for example, maybe they had a temporary pseudoinfection during a checkup that temporarily dragged down a little cluster of measurements. And step 3 is just pure measurement error, the random static and noise from the lab instruments. By separating trait, state, and noise, the model creates an unbelievably accurate picture of reality.
Let's move to Section 6, Estimation with ML and REM. Looking under the hood.
Okay, so we've built this beautiful, complex mathematical structure. But how does a computer actually solve it? Software uses specific estimation algorithms, and there are 2 primary ones you've got to know. First is maximum likelihood, or ML. The goal of ML is basically to find the mathematical values that put the absolute highest probability on the data we actually observed. It's fantastic when you want to run tests to compare different fixed effects, like, say, proving whether that F508 genetic mutation definitively impacts lung function over time. The second method is restricted maximum likelihood, or RML. RML is specifically designed to provide slightly less biased estimates of the variance components. In other words, it's better at handling all those random individual quirks we just talked about.
But listen up, because we need to talk about a crucial warning here. This is a classic statistical pitfall that ruins a lot of hard work. REML achieves its accuracy by correcting for the degrees of freedom in your fixed effects. And because of that restriction, you absolutely cannot use REML for likelihood ratio test if you're changing the fixed covariates between the models you're comparing. So if you want to compare a model that includes patient gender against a model that ignores gender, you must use ML. You really only want to use REML when your fixed effects are completely locked in place and you're entirely focused on refining the random variance.
Well, that brings us to our final thought for today's explainer. If linear mixed models give us the incredible power to elegantly isolate a disease's overarching trajectory from a single patient's individual traits and temporary states, what other messy datasets could this unlock for you? Whether you're tracking plant growth in different soils, economic trends across wild markets, or human health over decades, the real world is just full of uneven, clustered data. And now you've got the structured tools to make sense of the whole shebang. Thanks so much for joining me on The Explainer today. Keep learning, keep exploring, and we'll catch you next time.